Okay, good afternoon. I am Carlos Arteaga, co-chair of the San Antonio Breast Cancer Symposium for the AACR. Welcome to the special session, Overcoming the Big Obstacles and Finding Solutions for Breast Cancer Progress, which I will co-moderate with Dr. Angela DeMichele, professor at the University of Pennsylvania and the Abramson Cancer Center. For this forum, the program committee, Dr. DeMichele and I identified a panel of world-class experts spanning the spectrum of laboratory discovery, translational, clinical research, population science, cancer control, patient advocacy, and leadership in academic oncology and industry, and asked them to identify what they consider are the top obstacles to overcome for the next 5 to 10 years in order to make significant progress against breast cancer. We will start with a 10 to 12-minute introduction presentation where Dr. DeMichele will present the state of affairs in breast cancer, followed by discussion of the 6 main obstacles that these experts have identified. And then each of them will have a mini TED Talk on those obstacles and the transformative solutions they propose in order to get to the next level in the fight against breast cancer. These are the obstacles at the bottom, the ones that they have identified: prevention and early detection, underutilization of AI and big data, precision oncology, clinical trials process, cancer health disparities, and aligning incentives and funding for collaboration. At the end, we will poll the audience for what you think should be the top 3 priorities for progress in breast cancer in the next 5 to 10 years. We sincerely hope that this will be highly interactive. We would like a lot of questions from the audience. And I hope that this becomes part of the roadmap that we together can embark upon in the next few years, again, to continue the fight against this disease. So now I'll invite Dr. DeMichele to present some slides on the state of affairs in breast cancer today. Angie?
Thank you so much, Carlos, and welcome everyone back to San Antonio. Good to see everyone today. I'm going to just briefly, as Carlos mentioned, give you some data so that you can put into context the information that you're hearing today, to really drive home where we are now so that we can talk about where we need to go. So, of course, we must start with incidence and mortality. And what you see here are the data for incidence and those blue dots. And what's notable is that incidence is rising since falling around the beginning of this century when we had data about hormone replacement therapy, and we saw incidence drop. It has been steadily increasing since that time. Now, the good news is that mortality is decreasing, but it still remains high. And we think about this, obviously, I do here in the United States, but we really need to be thinking about this worldwide. And as you see on the far side, we still have a long way to go to tackle this problem of breast cancer incidence and the number of new cases, as well as the number of deaths. So 685,000 deaths per year, that was in 2020, too many. And if we look at where that incidence is on the rise, what's notable is that it's at the extremes of age. So those under the age of 50 and those over the age of 65 are in the groups that are rising, particularly amongst those under the age of 50, as you see there. And worldwide, we see the greatest increases in East Asia and the Pacific, the Middle East, North Africa, and Latin America. Now, in terms of prevention, early detection, and interception, we've long known about the risk factors that we see in these pink arrows. New data is emerging about risk factors in the green arrow: smoking, night shift work, sleep habits, hair products, chemicals, and importantly, social stressors and allosteric load. which really start to get at some of the social determinants of health that we'll talk about in this session. We've also had a new wave of innovation in AI and big data, but this has been primarily in the field of imaging. So what you can see here are a series of FDA approvals for AI-based software systems for early detection and diagnosis of breast cancer. We'll talk today about other applications of AI and what we need to do to bring things up to speed in other areas. In terms of biology, we've all long thought about this disease as one of these receptor subtypes, as you see on the left, and that those receptor subtypes confer specific 5-year survivals, and biologically we treat these diseases differently. But of course we know now that that's just the tip of the iceberg. In recent years, we have begun to unravel the molecular and genetic underpinnings of breast cancer. And as you can see here from the AACR Project Genie, the top 10 mutations that we see in breast cancer. We know breast cancer is a complex disease marked by both genomic and genetic abnormalities. And we also know that we can use these abnormalities to track breast cancer now in new ways. This is illustrating the use of liquid biopsy or circulating tumor DNA. And this really emerged with the first breakthrough status back in 2019. But you see rapidly now additional breakthrough designations, most recently in 2022, for a liquid biopsy test in early detection of MRD in early stage breast cancer. So this is really critical to our understanding the value of liquid biopsies in detection, evaluating response to treatment, detecting treatment resistance, understanding heterogeneity, and importantly, monitoring for minimal residual disease. We've also had a plethora of new drugs approved, and in the last decade, you can see 20 new therapeutics for breast cancer that received FDA approval in the United States, as well as a series of drugs on the bottom that are agnostic to tumor type. So we see an incredible explosion of new therapeutics that are available, but they are not universally available around the world. In addition, when we look at the patient populations that made up the clinical trials that led to these approvals, we see that there is a tremendous preponderance of Caucasian patients that made up those trials that truly limits our ability to interpret the results of those trials in people of color. And that's not surprising given, as you look here, at where those clinical trials have been located. So the numbers inside the countries represent the number of clinical trials that are funded on the left through government, on the right through industry, and you see that there is high concentration in the US, Europe, and China, with very few trials in several of those areas where I already told you that breast cancer is increasing. So we have geographical problems with where we're conducting trials in order to include a geographically representative group of patients in them. We also have tremendous disparities here in the United States. We know that there are various groups who do not have access to the latest technologies and therapeutics, and you can see that the NCI has noted the top groups that are experiencing disparities in cancer treatment that lead to low quality of life. And on the right, we have now begun to finally recognize and develop a conceptual model that enables us to finally begin to unravel the social determinants of health that are so important to understanding what is causing breast cancer and how we should best treat it in the individual patient and in the population from which that patient comes. Recognizing this, ASCO has created several tools. This is the research site self-assessment that enables research sites to identify opportunities to improve equity, diversity, and inclusion in clinical research. And there is also the Just Ask program, which enables clinicians and staff to assess and mitigate biases. And this is specifically in terms of screening and offering clinical trials. You can learn more about these really important programs at the website that's listed there on the slide. And finally, we need to align our incentives for funding, collaboration, training, and retention. We all know that we struggle with funding for research. The left-hand box shows the funding from NCI, which has been relatively flat over the last 10 years. That is supplemented by another $150 million in FY21 and 22 from the Department of Defense. And importantly, foundations play a major role in funding research that changes care. But that funding is subject to the vicissitudes of the markets and is not necessarily stable from year to year, as you can see. We also recognize that we have issues with our workforce. As you see on the left, when we move from medical students to cancer-focused residents to cancer-focused physicians, we see a decline in women, racial, and ethnic minorities entering cancer care amongst our trainees. And we have also seen a tremendous exodus of PhDs from academia to industry, which creates an imbalance within our ecosystem in terms of making progress. So with that background, to help you understand what we and this panel felt were the major problems that needed to be addressed, we identified these 6 obstacles. And what you can see here, I know Carlos introduced these obstacles to you a bit earlier, and we've asked 2 of our esteemed colleagues to start each of these off with a short talk about their philosophy and vision for how to overcome these obstacles, and we'll follow each of these with some questions and answers from the audience, as well as we hope some interaction amongst our panelists. So to start us off with obstacle number 1, prevention, early detection, and interception, I'd like to introduce Christine Emperzone, PhD, and Joan Brugge, PhD. And Christine, would you like to get us started?
Sure, thank you. So this may seem provocative coming from a chair of Department of Cancer Prevention and Control, but I think that overall prevention of breast cancer at a population level is unlikely. And one reason for it is because unlike, say, HPV infection and cervical cancer, smoking and lung cancer, as Dr. DeMichelle already showed, there's so many risk factors driving breast cancer. There's not one big one that if you eliminated, we'd really see breast cancer rates go down. Another barrier is that breast cancer is not one disease. It's very heterogeneous, and it's probably different exposures that are driving molecular alterations within those tumors. So they can't really, again, be targeted for prevention. So clearly, we need to focus in on those who are most at risk for prevention strategies and early detection. And there are effective tools for breast cancer risk assessment, but these are primarily for overall breast cancer. So when we're thinking of investment of resources for prevention or risk reduction, I think we really need to be able to identify those who are most at risk of poor prognosis, more aggressive breast cancers, such as ER-negative, high-grade, and triple-negative. And without delineation of who's at risk for poor prognosis breast cancer, broad use of personalized prediction tools may lead to overscreening and overdetection of tumors that are really low risk. So we need comprehensive subtype-specific models that include genetic susceptibility, high penetrance variants, and polygenic risk scores, which estimate the cumulative effects of multiple SNPs. These models will also need to factor in reproductive and lifestyle factors, and this is where I was thinking maybe AI is going to be able to handle this. Because, for example, we know that overall having children reduces risk of breast cancer, but this is only true for ER positive, and actually having children increases risk of ER negative breast cancer, but breastfeeding reduces that. So these factors, I think, really need to be incorporated into models. Then those at high risk of fatal breast cancer can be targeted for interventions such as earlier or more intensive screening, pharmaceutical approaches for prevention, and prophylaxis for those at highest risk. Now, practically speaking, implementation of strategies for individualized risk assessment is going to need a commitment and investment at a national level and of resources in the healthcare setting, which leads us to a final caveat, that risk stratification and personalized prevention may further exacerbate some health disparities. Not all women will have access to testing for risk or individualized risk models or follow-up for being at high risk. And some may not really find personalized risk assessment acceptable. So social and structural determinants of health will need to be addressed to make strides towards making personalized risk stratification and prevention available to all women.
Great, thank you, Dr. Amberzone. So Dr. Brugge, would you like to make your comments, please?
Okay, so I decided to, or I was asked to address the challenges relating to early detection and interception of breast cancer. And we were asked to think about what our vision would be for the next 10 to 20 years. So my vision in this area would be that we would have detection methods and biomarkers to identify expansion of the earliest premalignant lesions most likely to progress to cancer. And then once we can detect these, what I hope we'll have is safe and effective strategies to eliminate premalignant cells and prevent their progression to cancer. This is obviously a very tall order, but Carlos and Angelo said to be bold and provocative and to think about what it would take to achieve those goals. So for earlier detection, we obviously need to identify markers of early premalignant cells. And those markers would need to distinguish lesions most likely to progress to malignancy. And that's huge. There are huge obstacles in this. It's been very difficult, as you know, to predict DCIS, which are most likely to progress. But I'm hoping that new technologies that are available now will allow us to track single cells at both the DNA and RNA level and hope to be able to find cells that carry the earliest genetic lesions and the earliest state changes that can be identified and be able to track those with respect to their the outcome of patients that carry those. So markers of progression, we've been focusing on intrinsic pathways within the tumor cells, and now we understand that we really need to track the tumor microenvironment because it's very likely that influences from the tumor microenvironment are going to have a huge influence on whether cells with these genetic alterations are going to progress. So we need really significant, and we need more research to understand the cooperation between, or the interactions between the tumor cells, or the premalignant cells and the microenvironment. And then a huge need is what I would say is really significant technological advances to increase the sensitivity of detection methodologies. So this is likely going to require a transformative and highly novel approach because the fold increase in sensitivity that we need to detect these early expanding premalignant cells compared to detection of even small tumors is enormous. So then for interception, let's say we are successful in identifying these cells, then we need to identify the vulnerabilities of these premalignant lesions in order to eliminate them. And again, we need to identify factors in the tumor microenvironment that are promoting progression and figure out how to prevent that, the factors that promote prevention. So I think the routes to prevention could arise from strategies that are designed to flush out the reservoirs of altered cells that predispose to cancer. These could arise from aging, from hormone alterations, from inflammation, from sporadic large-scale chromosomal abnormalities, or chronic exposure to cellular stresses with inadequate DNA damage repair. What we look for are commonalities to these stress states so that we would be able to identify common states such that we can deal with the heterogeneity of the genetic interactions. We'll also need animal models to mimic these early events in order to test interventional strategies. So I propose a Manhattan-like brainstorming project bringing together people from all different areas, engineering, computation, biology, genetics, to develop strategies that we would have considered unimaginable in the past and figure out ways to achieve them.
Thank you. I love this bold thinking, and I love how complementary your two approaches are, and I think that we can all recognize that we can, the biologic and the epidemiologic are highly integrated, and we need to take both approaches. So let's open it up to comments or questions on this topic. This is now open, and please, I want to encourage everyone, you can send a question through your phone. If you don't want to go up to the microphone, you need to log into this session on the app, and at the bottom you will see something that says Q&A or polling. You go into Q&A, and that's where you can put your question. We also have questions coming in from those of you who are online, so welcome all of you. Let me start with one of those. So the first question is, what risk models do you recommend that have now been validated for risk assessment for the development? What do you think are the strongest models in terms of validation?
Well, I think the models in which tumor suppressors are knocked out are among the best models, because you don't have a driving oncogene that is such a dominant driver. So, for instance, the BRCA P53 alterations, I think, are really good models, because we know that that alteration in itself does not lead to a tumor immediately. It takes 8 months. So I think you can follow the progression. And so I think that's, I guess I would say that's probably the best model in my mind. But other tumor suppressive models could also be feasible.
We have a question at microphone 3. And please be sure to identify yourself before you ask your question.
I'm not Susan Tannenbaum, University, Connecticut. I just wanted to take pause about what you said about the difficulty in prevention of breast cancer. I think some of the things that we have access to, for instance, healthy diet and exercise, is something that impacts us in breast cancer care in a big way. And we spend very little time talking to our patients about this, and everyone in this room can impact that. In terms of social determinants of health, people have limited access to healthy foods, whole food, plant-based diet, and I'd love to hear what people have to say about things that are within our control that we can actually impact, and yet we sort of avoid that issue in discussion.
Yeah, I agree with you on that point, but I think of those factors more as being able to reduce risk, because when we think of prevention or eliminating breast cancer, there are so many different causes and paths to the disease. There have to be huge changes in so many risk factors to be able to eliminate breast cancer. But I agree with you. I think physical activity, we know now very clearly, is a great way to reduce risk. Some of the dietary factors have still not really been validated, but we recommend eat a diet high in vegetables and whole grains, reduced meat. But to me, those are really to reduce risk in a population
Prevent.
level.
Do any of our other panelists want to comment on this? Dr. Weiner.
Actually, I have a question, but first let me just ask for all the speakers up here, if you can speak directly into the microphone, we could all hear each other. So get right up to it. My question for the two of you, it actually relates to this problem we have of not being able to distinguish between fatal breast cancer and breast cancer that is never going to harm someone. And I'm interested in your roadmap as to how we're going to get there, so that in 10 years we're not going to be fussing over lesions that are irrelevant in someone's life, and at the same time being able to address those that really have the potential to take a life.
So I'll take a stab at that. I know that there is work going on. Laura Esserman, for quite a while, has been talking about interval cancers, and they just pop up in between screenings, and screenings missing those. And she's the PI of the Wisdom trial, which is testing different approaches to screening for early detection. I'm not really sure at a larger level, though. Joan, do you have?
Well, I think if you just look at what's going on in DCIS studies, that there, I think a lot of progress is being made in looking at interactions between the tumor cells and the tumor microenvironment. And my feeling is that we're going to get a lot of really meaningful answers from those studies, because inflammation and other alterations, like in fibroblasts, and there's secretion of lots of factors that promote the misbehavior of or progression of tumor cells, are, we know a lot more about that. So I guess I feel that these, this is like one road to follow. There's likely others as well. I'm curious if anybody else has.
Dr. Ellis.
I have a slightly different comment. I was thinking about the hereditary predisposed population with BRCA1 and 2 and others, where they have an extraordinarily high incidence of breast cancer. I was wondering what the group thought about making progress in that particular population. Of course, the question relates perhaps to the use of PARP inhibitors as preventative therapy, not just for women at risk for cancer, but also perhaps men at risk for prostate and other cancers. What would it take to get there, and what would be the ideal profile of a prevention agent for those patients?
You're allowed to phone a friend if you want to ask anybody else on the panel to
Is
to
Judy
answer.
Garber here? I mean, yeah, as a PhD, I don't have much to say about that, but I think that one of the most encouraging things I've heard about are the PARP1 specific inhibitors, which appear to have less toxicity. And I think that for those lesions in which there's been loss of heterozygosity, which we know occurs early, but before they've accumulated other changes, that loss of heterozygosity represents a vulnerability that's very attractive to pursue with a safe drug that will specifically target that instability.
Yeah, maybe I can ask Dr. Harbeck to comment on what a clinical trial would look like in that setting.
I think there is already trials ongoing with that regard, and I think that's a very important point. But when hearing this question, I thought to myself, well, why don't we start testing women earlier? And we have all these complicated criteria, but now that testing has become very inexpensive, I think we should make this available, because we do have a lot of very conventional options for prevention of breast cancer and ovarian cancer in those patients, provided they know about their test results.
Let's let our last comment be from our patient advocate, Ms. Patty Spears, who's down at the end there.
Yeah, so just bringing it back to the bigger picture, I think, as a patient looking at risk reduction and prevention of breast cancer, I think you really need to have some kind of perspective there is what's the end goal? Is catching it earlier going to be productive, and what's going to happen if you catch it early? Not just the act of catching it early is really cool, but what would you do in that case, right? And do you give these drugs that have toxicities to people? And so kind of getting that real world viewpoint, I think, is really important in this area.
Well, that was a great start. That was a great start. And I think, and some of these topics and questions will come back, because there are a lot of points of contact among these 6 obstacles that have been identified. The next obstacle pertains to the underutilization of artificial intelligence, big data, and the need of a digital transformation in the next 5 to 10 years. That topic will be addressed by Regina Barcilay. Dr. Barcilay is a School of Engineering Distinguished Professor for AI and Health at MIT, and Dr. Yeon-hee Park, who is Professor of Medicine at Sung Kyung Kwon University in Seoul. So we'll start with Dr. Barzilay.
Hey, is it working? Yes, great. So when I'm thinking about the biggest opportunity and challenge of applying AI to breast cancer, it's actually one task, personalization. Because all the problems we just had in this panel, detecting which tumor is going to be a fatal tumor, understanding how the patient reacts to the medicine, all of those problems, actually machine learning problems, where you have a lot of data, you have outcomes from the past, and you're trying to predict what will happen. And despite the fact that machine learning is really prevalent in all other areas of our life, in breast cancer, as I've seen even today during the conference, the most standard techniques based on statistics are still prevalent. And when I'm thinking about personalization, I'm really thinking about Amazon-style personalization, where every single click that you are making and product that you are buying or looking at determines what's to come. We don't have anything like that in breast cancer, where one looks at a number of expert-selected biomarkers and then stratify population into the groups. So this is, if you just now take existing treatments and existing information that you are collecting, and you are to bring their machine learning to do the matching from the patient's features to the outcome, we can already improve. But of course, there is immediate questions that comes in. What kind of data are you going to be using for it? And today we have a lot of data from clinical trials, retrospective data, that is severely underutilized. Radiology, as we've heard earlier, is just one area. There are so many more things we can do. And I also want to say that it's impossible actually to make this tool impactful for patients unless clinicians, people in this audience, and researchers who are not AI, who are really focused on patients, are becoming part of this development, and they are thinking how to develop clinical protocols and how to bring it to patients. So the second point that I wanted to make is about AI in really in kind of fundamental biology of cancer. And I was very lucky to hear the first session, and I loved Joanne's talk in the first session, many others. But it's really clear to me today that all the advances that happen in machine learning for biochemistry, AlphaFold, single-cell machine learning, all these other advancements are not really part today of the majority of techniques that are used to understand breast cancer. And my hope is that if we are going to take, the best of all the advancement in molecular modeling and drug discovery and combine it with the tremendous expertise in biology of the researchers in biology of breast cancer, I think, and I want to follow here the call, that we can really redefine the landscape of this disease and also make it personal for every patient in terms of detecting, in terms of treating, and in terms of mitigating the side effects. Thank you.
Dr. Park?
Thank you, Regina. It's very hard topic for me. So I have some big obstacle. We have some big obstacle in transformation from real-world data to biomedical big data. It's time for data science-based innovations. I don't think randomized control trial can be replaced with real-world data-driven, non-randomized observational studies. However, rigid and complex RCTs can be revised and upgraded as modernized clinical trial using big data and digital transformations. Actually, bureaucracy in governing clinical trial prevented from active enrollment and adherence. For example, expensive but relatively ineffective trial monitoring strategies such as checking source document all the times and making frequent site visits have reduced clinical trial accessibilities. These strategies definitely have been imposed by regulatory guidelines and enforced by many funders and regulatory inspectors. Developing guidance for improving specific aspects of clinical trial should be urgently needed and can be facilitated by digital transformation, including PRO-related applications and monitoring outcome wearable device, and especially real-time electronic data direct transfer system can allow efficient centralized monitoring of patient efficacy and safety and facility. And secondly, implementation into the non-randomized observation analysis from real-world data set, the EHR, registries, claimed and billed data. This strategy should be endorsed and supported by regulatory authorities. Obviously, these have two major prerequisites: overcoming discrepancy of accessibility of big data associated with heterogeneity of real-world data source. And secondly, keeping data integrity for digital transformation in clinical trial and data integration among healthcare information subdomains. Working smartly and collaboratively and embracing change are needed to evolve and deliver better data-driven decision-making. That's all for me.
Thank you, Dr. Park and Dr. Barcilay. I have a question to Dr. Barcilay. So I think it's my sense that artificial intelligence has been has had a better output in basic discovery and laboratory research than in clinical care or clinical trials transformation. And I wonder whether you agree with that, and if so, what are we missing? Is there the technology not ready? Is there resistance to its implementation? Is it too costly? Are we missing some key workforce in academic centers that we should expand now if we want to really transform digitally, the cancer research in breast cancer?
So thank you very much for the question. So first I want to note that there were really multiple successes of utilizing AI for running clinical trials, not in breast cancer. And I want to highlight one of them that was done in Janssen for identifying areas ahead of time where the COVID will show up, and they really demonstrated that it helped with recruitment and decrease the cost of trials. So there are multiple examples like that. And I think, as you correctly identify, one of the bigger challenges is the fact that the workforce is still not truly familiar with the techniques and opportunities. And even if we're looking at this meeting, AI is not really represented. So I think bringing to speed, the general clinician community is one way to go. The second big piece, which I think is really an issue for bringing AI to breast cancer, relates to data. On one hand, lots of data it's not easy to share today. It's not easy to bring it to AI providers. And also there is a lot of data that, for instance, is not even available. Like if we're thinking about young women who are likely to develop breast cancer, if you're thinking what kind of data you may want to have for developing these models, you need to have something which is available in European system where you have the whole medical history. You don't even have it easily in the United States. So really an investment on interchangeable data is crucially important. And I want to say that even the simplest data, like the corpus of mammograms, based on which you can not only learn who has cancer now, but predict who is going to get cancer. Or going back to Eric's question, who is going to get breast cancer that is going to kill them? We don't have this source. Now, each one of you in your institution have lots and lots of mammograms and the outcomes, but unfortunately, it is not it's not available to the broad community of researchers and people who can make this change. So,
yep. Thank you. We have a question in microphone number 3.
Sunil Badwe, Emory. The major advancements in the last decade has been with targeted therapy, and most of the data is still in very close private domains. How do we get that data out into the public domain and into the hands of people who can access this kind of data?
Anybody wants to take that?
So thank you for very important question, but I'm not the person who can answer the question. So accessibility of the big data is a very big still obstacle to resolve, but Anyway, there are some good examples in Europe and US, as I know. So there are some good examples from real-world data set from data registries. As I remember, the Belgium clinical data set has kind of a resolving the big registry and to high accessibility to big data.
Actually, I would really love to hear the clinicians like Dr. Weiner and the others that can tell us how one can get access to all the data from the trials. It's a very important question to all of us.
But I think Dr. Ellis had his hand raised first. But
Slightly preambles what I was going to say. But in any one clinical practice, less than 10% of patients will be going on clinical trials. Vast numbers of patients are receiving the standard of care and never get studied. So if you want to understand signaling inhibition, find a signaling inhibitor, give patients the inhibitor, study their tumors, look at the perturbations, and do it at scale, and do a massive hackathon, which could be, all working together globally to try and hack at some of these problems, like why are some patients resistant to chemotherapy, why don't TKIs work in all patients, et cetera, et cetera. I don't think we do a very good job of that at all. I don't think we need to rely on the clinical trial space to do an awful lot of that research.
I think that the question of clinical, I totally agree with you, but I think the question of clinical trial also relates to the question of utilizing retrospective data. I think it's a bigger general question that given how much data is collected about patients in breast cancer and how very little, and I'm telling you somebody who really tried very hard to get the data, and Eric knows about it, how challenging is to get it. And I'm really curious to hear the clinicians on this panel that can comment on how do you see solving this problem moving forward, because currently it resides in clinical centers where we don't really have computer scientists and AI researchers at the level that can really make sense out of this data in the best possible way.
Fabrice, you have something to
No, I think, Regina, you will be very disappointed on how clinical research organizations keep their data.
I know.
I mean, we have to be realistic. It's very difficult for people like you outside clinical research organizations that are very top-down organizations to get access to data. Okay. And second point, and I'm sorry, I'm going to argue a little bit with you. I would not like the audience to believe that everything is going to come from AI. I mean, we still need talented people working on experimental biology, coming with deductive, research. And last point, so I am from Europe, the continent you mentioned as a model. We also have our problem. We have GDPR, data protection, and we don't have so easily access to data, I can tell you.
I absolutely agree with you. I was not thinking that AI should supplement traditional biology. I think the power is combining the two. But I do think that if we see how AI changes many other areas and totally changes our life. I'm actually a breast cancer survivor. And I can tell you that I felt that it's really a humongous disservice to patients that many here want to change their outcomes, is the fact that many times this data is locked and is not available for doing science on it. So if we really want to bring digital transformation, which is the topic of this session, we as a community need to find the way to move it forward, rather than to accept the realities that it cannot be shared.
Yeah, well, thank you very much for that lively discussion. We need to move to the next obstacle, but clearly we need to hire 10 logarithm more of computational scientists at the clinical trials table. Thank you.
Great, all right. So we're moving on to obstacle number 3, precision oncology. Omics is not used widely enough. And to address this obstacle, we'll hear first from Dr. Fabrice André from the Gustave Roussy, and then from Dr. Matt Ellis from AstraZeneca. Dr. André.
Okay. Thank you. So for the first obstacle, I'm sorry because you asked us to be a little bit provocative and push a little bit the border. I think the biggest obstacle for genomic is that we all consider that breast cancer is a separate entity of all the other cancer. So what I mean here, I'm going to take the example of immunotherapeutics. In 2012, we had the information that PD-L1 expression predicts sensitivity to immunotherapeutics. The first trial in breast cancer was in 2018, meaning that because we consider that breast cancer is a separate entity, we lost 6 years of drug access. I did some calculation. It's around 100,000 women who did not get access to effective anti-PD-1 therapy because we consider that breast cancer is different than lung cancer, pancreatic cancer, colon cancer, etc. So I think the two solutions I am proposing for this, I think we all agree on the diagnosis because we lost 100,000 patients with access to immunotherapeutics. The first one is that I think molecular classification, when it's about metastatic cancer, should be the priority. And of course, it means that when it's about metastatic cancer, I think we should just remove breast cancer as an entity. We are talking about estrogen receptor positive, PIK3CA mutant cancer with I don't know which polymorphism. So I think that would be my first obstacle and solution. The second obstacle I see, and I think Matt will elaborate on that, is that more and more, when we do complex molecular characterization, we can identify a theoretical drug that probably could be useful for the patient, but we don't have this drug. So I believe more and more in the vision that, in the next 10 years, academic centers will make joint venture with biotech to create real personalized therapy, meaning we will create an antibody drug conjugate with I don't know which linker and which payload, so that I think is a solution to overcome one obstacle. And the last one is a question about reimbursement and payment of molecular testing. I think we should all be aware that molecular testing is a key solution to decrease healthcare disparities because you reduce the need for experts and you decrease treatment. And I think there is a need, to invent new mechanism of reimbursement of molecular testing, for example, based on the percentage of drug that has not been prescribed because you predict which patient benefits of which drug and do not benefit of another drug.
Well, thank you so much for inviting me. I often ask the students when I was a professor, why do patients die of breast cancer? And my answer was, because you don't know what's wrong with them. When we do work out what's wrong with an individual patient with breast cancer, say HER2 amplification, miracles can happen. But in many patients, we have no clue why that patient suffered a metastasis to the brain or to the liver, or why conventional chemotherapy didn't work, or why the endocrine therapy stopped working, etc., etc. We really do not understand what's wrong with most patients with breast cancer. So why don't we understand what's wrong with most patients with breast cancer? Because we're not looking in the right places. We do get partial answers from genomic analysis. We get some information for transcriptome analysis. But the problem with that is you're looking at the hard drive or the wiring without turning on the computer screen. The computer screen of life, and therefore of cancer, is composed of proteins and metabolites and modifications on proteins called post-translational modifications, with phosphorylation being an obvious example. We do not measure that routinely in any way in any of our cancer patients. So if we did, what might we learn? Obviously, this is a piece of work I didn't do with myself, but with my colleagues at the MIT, particularly Steve Carr. We asked, and the team in the proteomics unit there with funding from the NCI, the Clinical Proteomics Tumor Analysis Consortium, we ask a very simple question. Triple-negative breast cancer, a simple 2-drug regimen of carboplatin and taxotere, 6 cycles. Some patients win, some patients lose. Some patients have the pass-CR, and some patients didn't. We did DNA, RNA, protein, and phosphoprotein analysis of frozen biopsies before they start the therapy. And what do we discover? Dozens of biomarkers that are only visible in proteomics space. And on top of that, we discovered a brand new replication defect in triple-negative breast cancer that involves the deficiency of lagging strand synthesis components that instructs that triple-negative breast cancer to never respond to carboplatin. And replicating that finding in models and in subsequent data sets we haven't published yet. And that was only in 40 patients, because we turned on the computer screen of life. Imagine if we did that in hundreds of patients. Imagine we did that in hormone receptor positive disease. Imagine we try to work out why the standard of care Herceptin doesn't work, and cousins and daughters and sons of Herceptin doesn't work, which is why we have a poster here looking at 40601 neoadjuvant trastuzumab-based therapy, and found that the problem is not the tumor, it's the tumor microenvironment instructs that tumor to be resistant. So you can look at that poster too. So I think that's where I stand. We're not working hard enough to work out what's wrong with people, and we need to use next-gen's omics and computer science to sort that one out. Sorry, I went over time slightly.
So what I hear you both saying is really we need to be comprehensive in what we're collecting from patients, whether they're on a trial, whether it's standard of care, that biospecimens and annotation of treatment and everything we could record about that patient is what it's going to take for us to do a comprehensive assessment. And I guess I'd like to ask our patient advocate, Patty Spears, who I saw writing down there furiously, how you feel about that from the patient perspective, because I think aside from money, the major obstacle, of course, to all of this, how do you think patients would respond to that proposal?
Yeah, so it sounds like a lot. And I had written down when I had read this, I said, access should not be an afterthought. So we tend to do and do and do, and then we think about, how are we ever going to do this in the clinic, right? And how are we ever going to do that? And also in looking at, we get so focused on things as well. The scientific question is really, really intriguing and stuff, but then, you really have to look at the entire patient, right? So I think that there's a lot of things that we're missing when we're not looking at the entire patient, the comorbidities, the entire thing, as well as health lifestyles and different things, and kind of marrying that with the genomics and the proteomics type of thing. I think that's an area that we really don't do as well either.
Dr. Pierce.
And I also want to say, I agree with everything the panelists said, but we have to be very thoughtful in terms of what we do collect. I mean, we have to be pragmatic. If we want trials run in a large distribution of practices, not just at the major academic centers, they have to be trials that can be easily incorporated into the workflow so that clinicians can do it. So you can catch those rural patients. You can catch those patients that have difficulty coming into the major centers. So I think collecting tissue is important, but we have to be very thoughtful so that we don't overdo it and minimize collection from a more diverse group of patients.
Agreed. Dr. Harbeck.
There's more and more topics coming up to our clinical trial setup, so I'm looking forward to that. So I just want to add, I completely agree. I think we should ask patients much more. Fabrice said Europe is not so good with the data protection. Germany is even worse. So I think we should ask the patients what they want to have done with their data and then act on that and then have regulators tell us that. But I think with regard to the specimen collection, what I see in clinical trials is that they ask a lot for specimens, but there's nothing they give back to the patients and the clinician at the same time. So I think we have to look more to a win-win situation and not tell the individual patient whom we're asking to give up all the data and the specimens, well, we'll get to you once we know what to do with this. So I think we should, with all these new omics technologies, we should report back to the patients and their treating physicians what we can so they have an immediate benefit. Maybe they just want to know. Maybe there are factors that they can act on already. So I think it should be a win-win situation and not a one-way street.
Dr. Henry, you wanted to?
That is a great point. I think it needs to be more patient-centered. The whole idea of tissue biospecimen collection and things like that. It needs to be more patient-centered. You need to value that patient, and that's information, right? Going back, and I think we also rely a lot on clinical trials for a lot of this data. And, that's not getting every patient. So there really needs to be a way to bank biospecimens for future use on a global scale, on a more broad scale, so that we can have that information available. But I think, that seems to be stumbling blocks for some reason. But if you ask patients, doing what the patient wants, I think is really what we should do. And if I donate a specimen, I want you to use it to its fullest capacity that can help other patients.
No, I think, so I think, from my perspective, for each trial, you can have a different set of sample obtained, and in some trial, you don't need to collect any sample. In some other one, you need comprehensive assessment. The point I wanted to highlight is not so much this one, is why, when we know a genomic alteration provides sensitivity to a drug, why are we making 10 trials, each of them in a disease defined by WHO? It has absolutely nonsense. And I think there is no reason to test PARP inhibitor in breast cancer, then in ovarian cancer, then in prostate cancer, then in pancreatic cancer, then blah, 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 because it's extremely cruel for patients. Because if you don't have the right cancer defined by WHO, well, you don't have the right therapy. So there is a need for change on this. And I really believe that the breast cancer community should start integrate the concept that breast cancer should be diluted to other cancer when it's about metastatic setting.
Dr. Arteaga.
Yes, I have a question for Matt Ellis, who just challenged us to be a little more curious in the next decade. And you also suggest that we do have the tools to dial things up, that we could be a little more incisive in the investigation of the individual patient, not only in a trial, but also even undergoing standard of care. So I want you to comment on the fact that, and whether you agree or not, that why are we developing so many drugs that don't hit target? Why are we not incorporating omics early so we can triage drugs at the right time and not waste time, subject patients to unnecessary toxicity for things that are not going to work? How do we together, industry, academia, the community, work on again, avoiding that waste of effort and resources in developing drugs that don't hit target and go into 10 trials that may be unnecessary?
Well, of course, I spent 30 years in academia and,
That's why I'm asking you.
9 months in industry. But so I'm taking that mindset, obviously, to my current position. I think we don't measure targets very well, and so we're disappointed when you look for the interaction between that biomarker and the clinical outcomes. You're going to see examples of that possibly later this week. But I think overall, we're just not very good at measuring the right thing, and we're not very good at adopting new technologies rapidly that promise a better management. So, for example, everyone understands immunohistochemistry and all its foibles. Now you can measure the presence of a particular protein using mass spec with very high sensitivity and precision to the attomolar level. Why can't we develop a technique that would be called mass spec verified immunohistochemistry? So you get the expression variability and an absolute measure of the actual target present. So those kinds of experiments are going on. Finally, I just wanted to echo something that Paddy was saying, but also present a possible solution. So one possible solution to all this sort of consent issue and tissue use issue is something called ongoing digital consent. So the patient actually is digitally consented. You can see that they watched the appropriate video. They consented to the tissue analysis. Perhaps that's a screening sample, and the patient gets screened into a clinical trial. Great. But if he or she is not eligible for that trial because of the result of that analysis, that biospecimen still exists. It could be screened for a different therapeutic approach for the patient. But also, critically, because it's digital, it's easy for the patient also to withdraw consent. I don't want to be involved in this anymore. Click. So I think we need to use more techniques like that to reach that nirvana of what the patients want, what the physicians want, and what the drug developments want at the individual patient level. So just a suggestion.
Yeah, and I think a lot of patients don't understand the system and don't understand that when they say yes, they're only saying yes to one thing and not another thing. And so I think we need clarity in that as well when we
Sure.
consent patients. But I think that if you tell patients, up front what you want to do, and they would be happy. A lot would be happy to do it. Those that won't will not consent, and that's fine. But, I think that they don't realize all these little intricacies that happen that the institutions are constrained with, right? So I think communication is key there as well.
And Dr. Weiner, I know you had a comment you wanted to make.
Yeah. So sitting here, I'm struck that we have a problem that individual academic centers, individuals, individual drug companies, individual countries are all in their own little orbit. And then we try to coordinate it all. And just imagine the power for a minute if you had 100,000 diverse patients who were identified, from whom we obtained appropriate biologic samples, blood, tumor, what have you, stool, anything you want, that the patient is, of course, willing to provide. And you collected data. And you did this once prospectively, and we're done with it. I don't really believe you need more than 100,000 people. That's going to do it. But we just need to think a little bit bigger and a little bit out of the box here and not keep replicating the same things we've done for years and years.
I think that's a great way to end that particular obstacle, because I think that really brings together a lot of what we've heard already so far in terms of our need for bigger data, bigger biospecimen collections, and bigger approaches to these problems. So let's move on to our next obstacle. You want to take over?
Yeah, so the next obstacle deals with the clinical trials process and the need for innovation of something that may have been a little bit ossified for the last decade or so, at least in the opinion of some of our expert panelists. The discussant will be Dr. Nadia Harbeck, Director of the Breast Center at LMU University Hospital in Munich, and Eric Weiner, Director of the Smilo Cancer Center at Yale University. Dr. Harbeck?
Yeah, thank you. Eric and I sort of discussed this topic together. So I think we've heard in the other obstacles already that if you want to do something practice changing, you have to do a clinical trial. And in the future, I think we have to get used to the fact that we cannot answer all questions anymore by randomized clinical trials, because we've seen the changes in oncology worldwide, and therefore the better, of course, except for the limited resources, but we have limited patient collectives because the times of these one-size-fits-all trials are just long past, and now we have these very specific clinical questions, and we've seen decreasing mortality. We have more effective therapies in early breast cancer, which makes it difficult to establish new treatments in early breast cancer because patients fortunately do very well, so superiority trials from a trial design point are problematic. In the metastatic setting, hopefully in the future we'll have less metastatic breast cancer patients because of the progress we've made. And Angie pointed out we had 20 new registrations in the last 10 years, 8 of them in the last 2 years. So there is a lot of heterogeneity in the pre-treatments of patients, and obviously sitting next to Fabrice and Matt, there's also a lot of tumor biology heterogeneity we have to take into account. And lastly, due to the rapid progress, also guidelines have changed. We now have living guidelines both by ESMO and by ASCO, so maybe once a trial reads out, it's not so relevant anymore for the patients treated today. So what are the solutions? I think we've already talked about this. We need more real-world evidence and attach labels to that give them level of evidence that we can use in clinical practice, because we cannot possibly do a randomized trial for all of the issues we have at hand. And there is some very clever way using AI, for example, to recreate clinical trials by using real-world evidence. So it's like just somewhat like the biomarkers, where we saw that we didn't have enough tissue samples, so we created levels of evidence for biomarkers that include data that's already there in just prospectively planned analysis. And we can use that for the more mundane questions, but also for questions where we don't have enough patients. Like if you think about how to sequence therapies in the metastatic setting, there is just no way we can do a randomized trial for every patient with every single pretreatment. It's just not feasible. So we need real-world evidence, and we need to convince regulators to act upon cleverly planned real-world evidence analyses. You can also randomize them, you can use registries, and also change labels because of the data that comes out of there. And lastly, I think Eric said a very important point already. We need to collaborate more. We've already done this in the past beyond centers, borders, from this country for Atlantic Pacific, but we need to do this more, include the regulators, but also the payers and industry into that, because we don't have the resources to duplicate efforts anymore, and these small phase 2 hypothesis generating trials really don't help anybody if the conclusion is we need a phase 3 trial to back this up. And that could also be then the solution to save the resources to do for more targeted trials like Fabrice pointed out, but also more innovative trial designs. And that's when I hand over to Eric.
All right. So I'm basically going to make 2 points. And the first, not to steal any of the thunder from the next presenters, but relates to the issue of diversity in clinical trials. And you saw the data that Angie presented earlier showing the really inadequate participation of people of color in the vast majority of clinical trials. And we are simply not going to get answers to clinical trial questions that apply broadly if we don't include a diverse group of patients in those trials. And just to hammer this in a little more, as I think many of you know, if you're a Black American woman who is 20 years old, you have twice the chance of dying from breast cancer before the age of 50 compared to a white American woman. And that's for a lot of reasons, but we're certainly not going to fix it entirely if we don't include all patients in trials. Number 2, we need more innovative trial designs. We simply are not going to be able to answer everything with large randomized trials. We need 2 types of trials. We need trials that look at more and look at doing better, and we also need trials that look at backing off on therapy in situations where people are doing extraordinarily well. For those trials where we're trying to back off, oftentimes randomized trials are simply impossible. And doing single-arm trials in that situation and setting a target, for example, 98% of people alive and free of recurrence 5 years or 8 years or some number of years later, is a more than adequate endpoint because you can't do better. The one challenge with those trials, however, is it doesn't prove that a therapy is better than nothing. In some of those individuals, of course, nothing may be the right treatment, which gets back to my comment about breast cancers that are truly insignificant that we need to sort out. But then we have another challenge, which is that not all trials include all patients with all stages of disease. And so, for example, the RxPonder trial, which demonstrated quite clearly that in postmenopausal women with ER-positive, 1 to 3 positive breast cancer, that chemotherapy was of no value if a woman had a recurrence score of between 0 and 25. But what do we do with the woman who has 4 positive nodes, or 6 positive nodes, or 10 positive nodes, who has a recurrence score of 8 that someone obtained? And here, I think that it's much more challenging to do single-arm trials when the outcome is less pristine. That said, we don't want to be stuck in 1990 treatment paradigms for the rest of eternity. And trials may never be done that are sufficient randomized trials to answer the questions. And in those settings, we may also have to just be a little bit more comfortable with extrapolation, with single-arm studies, with registration-type studies, and we need to think carefully about how we move the field forward, even in patient groups that may not have been, strictly speaking, allowed in the clinical trial. And with that, I will end.
Thank you very much. Remember, we would like questions from the audience whenever there's an opportunity. I have a question for Eric. I agree with you that we need to think of new clinical trial models, particularly for those small genotypes. You know, for example, I can think that in 10 years, triple negative breast cancer will be broken into many subtypes, each identified by a genomic vulnerability, perhaps, you know, a mutation, an amplification, an RNA fusion, etc. So we have examples in lung cancer. The lung cancer people have done really well in, again, identifying treatments for small cohorts within non-small cell lung cancer universe, each with a specific therapy, okay? Why are we not doing some of that in breast cancer and still, again, doing a one-size-fits-all in many cases?
Well, I think that lung cancer has been blessed with many molecular abnormalities where there have been agents available to target those abnormalities. But I do think that in breast cancer, we're going to have to do that. And, you know, Fabrice was speaking about this earlier. And, you know, as we examine the molecular characteristics of individual tumors more and more, this is something that's going to come up. And as we define triple-negative breast cancer as 5, 6, 8, 10, 12 different diseases, it's going to happen. Now, whether in all of those cases we mount randomized trials or not, I don't have an answer to that. I think in many cases we will need to. But I think that we already have an example, which is patients who have germline BRCA1 and 2 mutations, where we know there is a specific drug that has a huge impact there, and in fact, where we've done trials in that setting.
Okay. Let's go to the, thank you. Let's go to the microphone number 4.
Hi there, my name is Nick McAndrew at UCLA. So I wanted to address something, Dr. Weiner, that you said. I think one of the challenges that you brought up is this idea that we need to be more collaborative, and I wanted to see what your thoughts were in terms of the major obstacles to actually rolling out a large collaborative prospective, even observational study, because it strikes me that in breast cancer, we are, you know, one of the, I guess, advantages of studying breast cancer is that it's a common disease, and that many individual centers can actually have enough numbers to actually do their own research, whereas I think what we should be doing is taking, taking a lead from some of our colleagues in rare disease networks. When my roommate in medical school came down with an extremely rare disease, not only did he become a world expert in it afterwards, but he developed a collaborative research network, and some of the innovations that they've had in being able to enroll patients even on tissue collection studies over the phone at different institutions are really innovative. And it strikes me that in breast cancer, we've never had to be so innovative because we have the capacity to do our own research at individual sites. And I just want to see what your thoughts were in terms of what those major obstacles are to actually rolling out large collaborative studies, and who should be leading it.
Is that me? Yeah. Oh, okay. So I think you've made an excellent point, and I think that to some extent it is because we just haven't needed them. And while you were speaking, I jotted down 4 possible reasons. One, of course, is it takes a lot of money, and so we have to identify the money to do that great big observational study. 2, I think there are academic issues which are going to be discussed in terms of people's need not for doing something other than team science and doing individual science. That will come up later. Number 3, a problem that isn't something that is not the source of the problem, which is the patients. And Patty may want to speak to that, but I don't think that this is an issue that our patients will not cooperate. I think this is something that we're in some ways denying them of. And finally, I just think it's been the tradition, as you noted. But, you know, I think that if we push it hard enough, it can happen.
Yeah. Microphone number 3.
Thank you. I'm Judith Bliss, the Institute of Cancer Research. So I want to just come back to what Eric and Nadia have been talking about. On one hand, clinical trials are seen as ossified, and real-world data is the sort of, the new horizons. Yet let's just come back to think about the data. So in a trial, again, as a community, we've sort of become a little bit fixated on checking every single bit of the quality of the data and completeness, all the auditing and monitoring. And that's one of the things that has led to a lot of these trials being very costly and very time-consuming. And yet with real-world data, that's data that's not collected prospectively for research, and we seem to be, whilst accepting that there are quality issues, at the same time we just accept that data. So, there's common sense that says pull the two together, and let's find a way of doing big pragmatic trials, but doing them much more cheaply by accepting some of the compromises that we are doing when we consider the real-world data, and not looking at the sort of all the fixation on monitoring and audit that has done to date.
Thank you. Dr. Harbeck, you want to address that?
Yeah, Jules, I think you're completely right. I think we've become over-bureaucratic with the way we're doing these industry-driven trials and also academic trials, to a point that we cannot afford to do these academic trials anymore. And I think a little bit more real-world data quality is good enough to answer some of the clinical questions. And in some of the trials, you have to be very specific. Like if you go down to omics or so, you have to make sure that the testing systems are state-of-the-art and so forth. So I think we have to find a compromise, and I think we have to sit together also with regulatory agencies, because a lot of these requirements don't come from academia or industry, but they come from the regulatory agencies, and we just have to tone this down. And we've already started doing that, because like with these new drugs that were developed, like antibody-drug conjugates, we have the pivotal trials. The results are so fantastic that they immediately come into clinical practice, but we lack so much evidence on patient collectives, on safety in the clinical practice in patients that are not so well selected specifically for trials. So regulators have come to ask for these registries in order to move forward. And with that comes also less quality of the data, because we don't check every data point 3 times before we enter it into the ECRF. So I think you're right. I think this comes back to the overriding topic of collaboration with regulators, with payers, and with industry. Matt, do you want to add to that from an industry point of view?
Yes. AstraZeneca and other companies do similar things. And actually, Eric's a little bit involved in this, something called Partners of Choice. So basically, getting together a large consortium of different academic centers, doing some of these studies, doing joint hackathons where we're collaboratively looking at that data, and then putting it in the public domain. I think that's a very important principle, because the reasons why pharmaceutical companies are not always successful are the same reasons why individual patients are not always successfully treated. We don't understand what we're treating adequately. So I think building academic industry partnerships is one of the reasons I went to AstraZeneca, and I'm all ears for people who want to join in.
Yep.
But this is something we're doing in 2023.
Okay, thank you. Microphone number 2, please.
Beth Emery from the Alamo Breast Cancer Foundation. I have a question about COVID and what happened in the development of those vaccines, and whether we learned anything and how we were able to develop them, test them, and put them in people's arms so quickly that we might be able to adapt into improving clinical trials generally.
Who wants to take that one?
That's for anybody out here.
I mean, I think it's, I'll just start and say, because I think it was the singularity of purpose when we had a problem and everybody was working on the same problem at the same time. It shows you the power that you can generate to solve a problem.
I can tell you, being director of a comprehensive cancer center, that even though the lessons from COVID allowed us to only very transiently stop accrual to trials, by implementation of those lessons from the pandemic led us to completely reactivate clinical trial accrual. So there were lessons learned. Now, we may have learned there may be other things that we can do in the future using RNA vaccines for breast cancer, but that's not the topic of this discussion right now.
Thank you.
Does anyone want to talk about the innovations in running trials that we were allowed to adopt during COVID to keep those trials going? And whether we're going to continue those now that it's somewhat past?
We are continuing them.
The decentralization of consent, electronic consents, and being able to have blood drawn locally rather than at the center, being able to space out visits, all of those were innovations that came about of necessity during COVID that we are now trying to maintain within the clinical trial infrastructure.
Fabrice, one last comment so we can go to the next obstacle. Fabrice?
Yeah, I'm going to be short, then Nadia can complement. I think I understand the need for collaboration, infrastructure-like comprehensive assessment of everything, but the reality is that the most important thing, we don't know yet what it's going to be. So I'll be very careful of, making too many top-down cohorts where we believe that we know what we are supposed to obtain. The best example, or one example, is microbiome. 5 years ago, nobody would have collected any feces, and now it's all about this because some people came and said, oh, we need to add. So I think it's important to have all this comprehensive cohort perspective if we are capable to aggregate new dimension of biology and new specimen.
Carlos, 15 seconds.
Please.
So I think the point that Judith raised is huge. And clinical trials have become totally obsessive compulsive. And the contrast of using real world data at times with garbage real world data, not always, but sometimes, and that contrast with the obsessive compulsive behavior around trials is crazy. And we have to make them easier. Like, as you did, I'm sorry, Nadia, and I just want to say, what Angie is talking about, what we did during COVID, of course, we should be able to do all the time.
Yeah, there is no need to frown upon real-world data because that's the data we treat our patients with every day and they get the best possible care from us. So I think what we need to do is find mechanisms how we can sort of create levels of evidence from real-world data that makes us change guidelines, change treatment algorithms and so forth. But real-world data is the foundation of what we do every day. We just have to find a sort of a way to deal with it in clinical trial design.
I was going to say what Dr. Harback just said. I think we keep comparing real-world data with clinical trial data. It doesn't compare. It's not the same thing. And so what can we learn from patients treated in the real world? And we need to learn from patients in the real world so we can better inform the trials and upscale our trials and do better trials. And so my ideas for what Eric said about de-escalation studies, we aren't doing it right if we're doing de-escalation studies. My idea is we never need them again because we do it right the first time.
Right. Okay. All right, so we're going to move on. That was a great discussion. And our next obstacle, cancer health disparities, is truly cross-cutting here over really all of the different obstacles we've talked about. And so to lead us in that discussion, we have Dr. Lori Pierce, who's a professor and vice provost for academic affairs at the University of Michigan, and Patty Spears, who is the lead in the Patient Advocates Research Council at the University of North Carolina at Chapel Hill. So Dr. Pierce, lead us off.
So thank you. So this is a huge topic, and we've heard quite a bit from previous speakers that apply to cancer health disparities. And certainly when Dr. DeMichel initially presented the incidence and survival data, and you saw, of course, that survivals have been improving with breast cancer, but as everyone knows, they have not been improving to anywhere near the same degree for women of color. So there's much work that needs to be done. So if we're focusing on solutions to cancer health disparities, first, we have to acknowledge that there is racism. There's racism broadly in society, and there's clearly racism in medicine. And I think once we acknowledge that, then we're willing to look at things in our practices of what can we do or what is wrong here that we need to change. And we can think very broadly and look at things that perhaps disadvantage one population over another. And a great example is the Clinical Treatment Act. This was something that has now been passed into law in the U.S., but it was a fact that patients who had Medicaid didn't have routine care costs covered when they participated in a clinical trial. Patients who had private insurance, it was covered. Those who had Medicare was covered, but Medicaid patients, it was not covered. So it was a disincentive for people who don't have money to participate in clinical trials. Well, thanks to people realizing that this was a policy that existed for many years and was disadvantaging Medicaid patients, this has now been changed. So I think coming to the realization that there is racism and we have to act on that is very important. And I think we also need to look at our own selves. You know, we all have biases and implicit biases, and we have to realize what they are, and we have to mitigate those biases. And Dr. DiMichele talked about one of the tools that we have for implicit bias that was created through ASCO and ACCC. And tools like that are very important for acknowledging and helping to mitigate our implicit biases. But you also can't look at this as a one and done kind of thing. It's like, okay, I've done my implicit bias training, I'm good. It's an iterative process because the more you do it, the more you really take home those messages and you bring change into how you interact with your patients. I think another point is we need to be better listeners to our patients and really understand the barriers that are facing them. We've talked about social determinants of health, but what barriers are they facing? You know, we as clinicians were all taught how to take a history when we were in medical school, but we really weren't taught how to take a social determinants of health history. So until you really know what the barriers are that your patients are facing, we have all of these amazing therapies, but if they don't have the ability to come in because they can't get off from work, they don't have transportation or whatever, then these advances don't help a wide group of our patients. So we need to be better listeners. And I think we also need to look at institutional processes. We've talked a lot about clinical trials and the importance of clinical trials. Well, in clinical trials, as we heard from Dr. Weiner, they need to be available to a wide range of patients of races and ethnic groups. And every institution, every practice has different barriers. The barriers at my institution, University of Michigan, are probably not the same as the barriers at your institution. And so there are tools, there are site self-assessment tools that allow us to look and see what the barriers are, where we're losing people, including patients of color, going into clinical trials. I always give the example. Let's say 100 African American cancer patients came into your institution in the past 3 months, and 3 went on a clinical trial. Well, what happened to the other 97? Did we not have the right trials? How did we lose these patients? And that's what we need to really, really carefully look at our institutions to see where we are losing a very large percentage of patients, and particularly patients of color. And then finally, I want to just comment on the comment that came from a person at the microphone earlier on about prevention. I think you were spot on. We as clinicians need to talk more to our patients about changing lifestyles, about not smoking, moderate to no drinking, increasing exercise, decreasing all those kinds of things. That's easy to do. Talk is cheap, right? We can do that with all of our patients, and that can go a long way in terms of preventing a large number of cancers.
So I think that so what I did was I actually asked other patient advocates for their opinion about what we were going to talk about, so it wouldn't just be my opinion up here. But I think health disparities was one of those things that came across under every category that they talked about. You know, one of the categories was treatment, detection, but access was really one of the topics that came up a lot, especially in health disparities. So I'm going to talk about 3 different topics, 2 to do with clinical trials and broadening eligibility and decentralization, and then one in just clinical trials and clinical care and navigation. So the broadening eligibility criteria, or modernizing the eligibility criteria, there has been activity in that area, but as a patient advocate sitting on a lot of trial development teams, I don't think the message is coming across. And in breast cancer, just allowing men into trials allows access for men to get onto trials. You might not have that many men, but think about the patient first. Patients that are male have no access, so every trial should accept male, unless there's a scientific reason not to have them on or a safety reason. I think all the eligibility criteria are really hard to manage as a patient. I tried to get into a clinical trial. It took me 3 tries. I wasn't eligible because of eligibility criteria of blood counts, HLA typing, things like that. So we need to really take that into account of who's missing from those studies. Decentralization, it was talked about with the COVID example, is another area, shipping, oral medications directly to patients that are on clinical trials. That would really save us a lot. As patients, we wouldn't have to travel so far. Having my blood test done a half mile from my house is a lot better than me going all the way to UNC to get it done, and things like that. Just little things like that I think would be really good to kind of think of the patient first. Right now, clinical trials are very trial-centric. We develop the trial and then we look for who the patients are out there, and I think it should be patients first. What do the patients need? And then develop the trials and look at the patient and say, what do they qualify for and what do they need? And it would be kind of flipping it. And then navigation, that came up on several sessions as well. And I think, some patients, especially when you're talking about diverse populations, rural populations, if they have somebody to guide them through the system, it's a lot easier. Even as an educated patient advocate being diagnosed with a different cancer, it's very daunting out there. I felt very unprepared, and so having someone to talk to about it is really important. And if you have that person be like the patient, I think that's really good. Me as a patient, I liked my doctor at one point because we were the same age, right? Because age matters, I think, and all of this matters. So I think that navigation programs through clinical trials or clinical care would really help with that access issue to quality care and access to clinical trials.
Thank you. So I don't see any questions at the microphone, so let me ask a question. I know you brought up the issue earlier, Dr. Pierce, about, okay, we've got 100 patients, African-American patients come in and only 3 go on the trial. How do we start and continue that conversation with the other 97 to understand what the barriers are? I mean, we've acknowledged that they're there. We acknowledge there are barriers. We acknowledge that we are not accommodating them. But where do you see us going in terms of whose responsibility is that? What is the skill set that we need?
Yeah, so as I mentioned, the barriers vary by where you are. We all have different systems, and some aspects of certain systems work and other aspects don't. And so there are site self-assessment tools, and ASCO and ACCC built one that's now available, and you showed it on your slide, that take you through the process of when a patient comes into your center, and you follow that process, and you look at your responses for what your institution has or does not have. And that gives you an indication for where your potential challenges are and what needs to be changed. It could be something like you need better navigation for the patients. It could be that you didn't ask them to go on clinical trials, which is a big issue, and we'll talk about that. It could be that you don't have the right trials at your institution for the patients that you see. So there are many touch points, and I think it takes time to go through this tool. But if we're willing to put that time in on the front end, it can make a significant difference in terms of being able to intelligently address the challenges at your institution.
Thank you. Dr. Ellis, did you want to comment on this issue?
Yes, I do, actually. I spent 13 years at Washington University in St. Louis, and as you know, St. Louis is one of the more segregated cities in the United States. And there was an interesting experiment happened, which is the city hospital closed. And so then there was a contract with Washington University and the Siteman Cancer Center to look after the underserved, actually at the university hospital. And subsequent to that, actually, we over-accrued relative to the population African-American patients. What that taught me is where you're cared for. It's nothing to do with the lack of willingness of African-Americans to participate in clinical trials, quite the opposite. It's the question of access to clinical trials, access to great care, and not creating, let's say, healthcare systems that segregate patients into places where they're not getting access.
That is an excellent point and an excellent observation, and I'll talk about this later, but we know that there are data that clearly show that patients will go on trials at the same rate, clinical trials, whether they're black, white, Asian, or whatever. You have to be able to have them in your system and ask them to go on trials. So I think that is an excellent point.
And Dr. Weiner?
And I think it's been shown too that we get a lot as patients. Well, the patients don't want to go on trials. No, the system is really keeping them out of the studies. The trials are not getting to community sites. We need trials that are less complex, that can benefit patients and can benefit the whole system, but that can get out to the community sites. Something as easy as that, but it's not the patient problem. It's actually more of a system problem than a patient problem.
Let's go. I'd like to get this question from microphone 3.
Hello. Congratulations to all the panel and the presentations to talk about these provocative hot topics. My name is Juan Carlos Amame. I'm a medical oncologist from Lima, Peru. But I made my training in Europe. And one of the biggest obstacles that I figure out when I return to my country is that we do not have the number of trials that we would like. And this situation in LATAM is very common. Where in LATAM we see a lot of big disparities and barriers in access. How can we do to shorten these barriers and to perform maybe some kind of alternative like real-world evidence or this stuff? Thank you for your answer.
Who wants to take that? Anyone?
Yeah, I can. Well, maybe I'm sorry, maybe I will not answer your question exactly, but it gives me the opportunity to. I fully agree with your point. I think part of the problem in trials is that 95% of the data that are acquired will never be used, and that costs a huge amount of money. And I suspect it's part of the reason why some trials cannot be done, for example, in your center. So I fully agree with you. We need to simplify clinical trials, move to what we call real-life trials, and then because of that, we will have more centers to run the trials. The other thing is that we underexplore the very short duration of highly effective drugs. Example, immunotherapeutics in MSI cancer, for example. This kind of trial should be available everywhere in the world because it's all about reducing resources to cure cancer and improving access to drugs. So I fully align with you. I suspect part of the problem is the amount of data we want to acquire that will never be used, plus the core funding of clinical research organization.
Anybody else want to comment on that question? Yes, Dr. Harbeck.
Yeah, I think that's you're raising an important point that, and Angie showed it, that some parts of the world there are just not enough clinical trials because maybe they're not so attractive to industry, or unfortunately in the countries where the revenues are greatest early after registration, also a lot of the trials are being done there. I think that's a fact, and I think what we can do to counteract this is increase the quality of clinical trials in countries by sort of creating central networks, like you're from Peru, I understood, so create central networks, try to get easy access to the centers, increase the speed of implementation of clinical trials around these centers, and I think that will be attractive then to do clinical trials. And also you as a community, you can ask your own questions and try to do the clinical trials that you feel, but I think it's all about collaboration, creating networks and being fast in responding to either industry trials or creating your own research questions.
First, Dr. Pierce, then Dr. Arteaga.
Yeah, I just want to add one other point, and you know, your comment was from your country. It also applies to rural areas in certain parts of this country as well. And so, again, it gets back to the comment we made about being more pragmatic about our trials, making sure that if you only have a limited ability to run certain trials, that you have the right trials that you have for your patient population, and that you have trials that can be more easily incorporated into the workflow. I mean, up to 20% of our patients in the U.S. live in very sparsely, very sparse areas where access is a problem. So if you're going to treat them closer to home, you have to have trials that are easier to run. And so I think your point is well taken, even for the U.S.
Carlos, did you want to?
Yeah, no, I wanted to, this problem, this issue that you bring about these global disparities in clinical trials access, I think is very important. It has to go hand in hand with a big project on training on clinical research and translational research talent at those in these countries like yours. And this is where ASCO, AACR, ESMO can help out, I think, because I think that there's going to be a need of leadership at those sites, clinical leadership, like you are, and we to expand that further.
Well, I think that is a perfect segue into our final obstacle. Carlos, do you want to take us home here?
Angie.
Oh, we have another
Angie.
question? Yes? Oh,
So,
Patty? Did you want to speak and then?
No, you were going to do the next one is, yeah, we have another
But, Patty, go ahead and make your comment.
So I was just kind of thinking about what Nadia said about, RCTs, the, randomized clinical trials. If we didn't do those as often, if that would simplify the trials, not only from a patient perspective, but also from an institutional perspective and a physician bias perspective, if that might be helpful as well.
Should we take this last question? Microphone 3, please go ahead.
Hi, Anna Maria Storniolo from Indiana. This discussion leads me to wonder, again, it's about collaboration, but about those 97 people that we're not getting. And I wonder if we could learn from what we did during COVID and even get more creative. Any one center cannot open all trials. But if we knew the inclusion criteria for the trials close to us, is there a way that we could share trials, if you will, and potentially treat, maybe not on phase 1, et cetera, but share trials and treat people at our center on a trial that really is open somewhere else? Because again, we can't possibly open everything at all of our given trials.
Excellent point. Thank you so much. I think we'll go on to our last obstacle.
Yeah, the last obstacle before the polling is number 6, and it's aligning incentives and funding for collaboration, training, and retention, discussed by Fabrice André, Dr. John Brugge, and Dr. Laurie Pierce. Dr. André?
Yes, so I'm going to start with the funding, and I think then it will be other topics. So for the funding, I will just report our own doctrine and how we work in the foundation, our work. So our doctrine is very clear, is that funding should go to talent, meaning so everything should be done to give the talent the resource, first to be happy, and then to make their project. So what it means, I think, that first funding agency, they should really stop funding organizations that have too much administrative burden. This is a real problem. We give money, the money goes into administration, etc., people who have taken the control of research organization. So I think it's really important that funding agency monitor the level of administrative complexity and stop funding organizations who do no longer research but are only doing administration. The second thing is about in terms of program. So program, I think we should give funding to teams rather than organization. And then organization will fight and will develop the best infrastructure to attract all these teams. The point number 3 is that I think more and more we are funding projects that are telling us how cancer is complex. But in reality, there are a lot of teams working on finding solutions. So personally, I think funding should go more on finding solutions than project telling us how cancer is complex disease without any solution. And here I'm talking about biotechnologies, new therapy, new diagnostic, etc. The last point, I think, is about interdisciplinarity. So more and more, we are trying to fund projects that link teams in the space of biology, biomedicine, with teams doing science totally outside all this field, because it has been reported that this link is more likely to create breakthrough discoveries. And last point is the one we just discussed with our colleagues from Peru. I think charities should stop funding clinical research organizations that collect huge amount of data for nothing, or spend high amount of core funding in inflated organizations that are not effective. I mean, philanthropy, the money is given by donors who want to cure cancer. Money should not go to big administrative organizations who are making life of everybody very difficult.
Thank you, Fabrice. Dr. Brugge?
Okay, so I'm taking on the challenge of the academic brain drain. And I actually have more to say about the symptoms of the brain drain and the challenges that it presents than the treatment or the solutions. Because it's really impossible to deal with such an incredibly complex topic in 3 minutes. However, I feel that the situation has become more acute, and government institutions and scientific community need to take it very seriously. So one can view this at many levels. So at the faculty level, we see migration of some of the top physician scientists in cancer going to industry. This leaves significant leadership gaps in many institutions. We see this more in clinical areas than in basic science. In my department of cell biology, I don't think, besides myself, I've ever had someone go to industry. But also graduating medical student and resident fellows are less likely to choose a research track these days. And at the postdoc level, it's just there's been a dramatic transition. So we see many fewer graduate students choosing academic postdocs when they finish their thesis. And then at the research staff level, it's become more and more of a significant problem because the salary disparities in industry make hiring and retention extremely difficult. So this is, and also this is most acute for computational scientists who are in greatest demand in academic research, yet the most difficult to retain. It's so nice to have Regina and Shunsi with us and maintaining their role in academic science. So while we're very happy for our industry colleagues who are so successful in attracting and training the highest quality investigators, the situation is threatening progress in academics. So since we first started talking about this session, I've been talking with a lot of students and postdocs and also reading articles on the brain drain. And it seems that, if I could summarize, that the realities of academic research, so the poor success in securing academic jobs and securing grant funding is really discouraging PhD scientists from entering academics. This also applies to MD physician scientists. They see the risks of getting an academic job too high, and then if they are successful, they see enormous time devoted to securing grant funding instead of research. Also, students tend to be older when they finish their PhDs or MDs, MD PhDs, and they're tired of living on minimal wages. They're ready to start a family, which is extremely difficult on postdoc wages. So they reason that if they're ultimately going to end up not getting a job in industry, why not skip the postdoc? I think physician scientists also fear that the demands to straddle their clinical responsibility with demands to stay on top of a research ladder and secure funding may be too great. Also, computational scientists can engage in interesting projects in industry without the need to get grants and deal with the less than stable academic positions. So overall, the enormous benefits that we all have experienced through our careers and still to some extent today, I think they're being overshadowed by some of these issues and causing trainees to think about other options. So there are no simple solutions that I can bullet in this lightning session, and we definitely need different solutions for different components of the community. I feel like we need societies like AACR, ASCO to work together with government institutions and other funding agencies to find broad solutions. Some of this can be addressed in funding, and that's a significant issue, but some may require more serious restructuring. Clearly, more funding will bring higher salaries for postdocs and research staffs that would partially help them from being lured to industry. If grants could be larger, it would avoid the necessity of each lab carrying so many different grants, especially when funding is so low. But I think other solutions are required. One of the things that I've talked with my computational colleagues is that often the academic departments don't provide standard tenure track positions for computational scientists. They're kind of considered a different class. We need to have more stable tenure track positions. And also, I think that one thing we should think about is a more stable career structure for non-PhD research scientists that would allow them to stay in industry and salaries to support that, so that we can keep the workforce active when there's diminishing postdoc entry into the field. So there's lots of solutions that are needed, and I think that we really need to put this as a high priority in the societies and bringing these to governments and other funding agencies.
Thank you. Dr. Pierce.
So I'm going to continue the discussion about brain drain and what can universities do to keep great scientists and clinicians in the universities. And I'm actually going to have practical steps. I've thought a lot about this in my role as vice provost. We need to have time in the universities to be successful. If you're on the tenure track, you need to have a period of time where you actually can be successful if tenure is what you want to achieve. And so I think we all need to look at what the time to tenure is at your institution, and is it adequate? Because we know it takes time to get funding. If you look at some of the more recent numbers, the first R01 for MDs and MD PhDs is at age 46 on average. It takes time to get that first R01. And so we need time in the tenure system, and we also need to think outside the box, and perhaps an R01 is not necessary to be promoted. There are other funding sources that should be considered. We shouldn't be so rigid that it has to be an R01. And Dr. Weiner brought this up earlier about team science. I think we all need to rally behind the importance of team science. You know, with regard to promotion, it's in the past it's always been I did this, I did this, I did this. That's not the way of the world. That's not the way that we are successful in making major contributions to the improvement of lives of our patients with cancer. It takes a team approach. And we need in universities to recognize and reward team science. The currency for promotion has always been grant funding and publications, and that's not going to change, but we can also augment that to recognize the importance of team science. I'm very happy to say I led a committee at my university this past year about incorporating team science in our promotion criteria, and it was accepted. We can do this. We need to promote team science. We also need to promote and reward entrepreneurial initiatives. Thinking outside the box, change agents, that can bring about true change. We need to reward our entrepreneurs. You know, many of the creative ideas that come from faculty who think outside the box can truly impact society and support the mission of universities, and that is to improve the lives of change the world, right? Improve the lives of everyone. And so there is a multi-institution coalition that is working at many universities to bring some of the entrepreneurial insights into universities to make them more aware that that's something that should also be rewarded. So again, we need to think outside the box. We need to to expand the currency for success at our institutions. And it can be done. I know institutions can take a while, but it can happen. And then finally, we need to have more family-friendly policies so we have a better work-life balance. You know, if you go to industry, you often have paid maternity leave, paid parental leaves. You can have that also in academics. But we have to make a work environment that is more conducive to being able to have a family life, but yet be successful. So there's a lot to university life to enjoy. Our careers are very stimulating. We get to interact with the next generation of learners, collaborate widely, interact with people like in the arts and humanities and social sciences. There's a whole world out there, and there's much to be to enjoy in university life. We just need to make our lives as scientists more acceptable.
Thank you, Lori. Because of the time, we're going to have to go straight to the poll. This issue of the brain drain and all that, we have to continue that conversation later, but let's go to the poll because we only have 3 minutes left.
Great. Okay, so you've all spent the last 2 hours, or many of you have spent the last 2 hours hearing about these big obstacles. We've moved from prevention, early detection, and interception to underutilization of AI and big data, digital transformation, precision oncology, clinical trials process, cancer health disparities, and aligning incentives and funding. So now it's your turn. You've heard from our all-star panel of solutions to many of these, but we want to know what you think. What do you think should be the priorities in terms of tackling these big obstacles? So we'd like to just take this last couple of minutes and have you vote. So your first job is to vote for what you think is the number one, most important obstacle for us as a field to address. You can find the opportunity to vote right if you tap within the app at the bottom of the screen. There is a place that's for Q&A and polling, and if you go into that, you can go ahead and just vote for your number one obstacle you think we should be focused upon. We're going to go to that. So you've got 9 seconds to vote, and then we'll see our results. All right, so we didn't get too many responses. It's going to take people a little while to get used to this polling technology, I think. But I think what we see that there is a lot of interest in aligning incentives, clinical trials process, and prevention. Can we next go to what you think is your second most important obstacle to overcome? So you get to vote again. What's number 2? We're going to get your top 3. So we want your top 3. So number 2.
It's not coming up. Q&A polling. What's happened?
Oh.
The system's not working. It just doesn't come up.
It isn't coming up for you to vote?
Maybe he cannot vote.
We think we should. Let's see what happens here. Not getting a chance to vote? All right, well, we got a few more responses, so you can see where the second most important obstacle is. Now give us your third. We'll average these for our purposes later on. Go ahead and give us your third most important obstacle. Okay.
So what was number 1?
I think so. Clinical trials and hit one more. All right. Great. Okay, so I think we're getting a handle on where people's priorities are, and I think with that, we would both Dr. Arteaga and I would like to thank our esteemed and all-star panel for all of your insights today on these big obstacles, and all of you for joining us. Let's give them a big hand. Thank you so much.